is episode 303 of the Stem Cell Podcast, Organoid Models of Cancer, with Dr. Alice Soragni. Hey, everybody. We are Doctors Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, as our guest, we have Dr. Alice Soragni, who's from UCLA, and she's on the podcast to talk about her research on protein aggregation and cancer organoid models. We've also got our usual roundup of recent highlights and stem cell news coming right up.
But first, interested in exploring the applications of organoid cultures? Get a free wall chart showing how organoids are used as model systems to study infectious disease, cancer, congenital disorders, and tissue regeneration. To learn more, visit stemcell.com/organoids-modeling-wall chart.
All right, guys, I want to set it off today with a little bit of clarification on the last episode's roundup. It was communicated to us that I dropped the ball a little bit in relaying the details of the De Silva paper, which Arun went back to back. I did the De Silva, he did the Jakob Hanna paper, and very similar stories, but I kind of missed a critical element, which is that they actually incorporated the roller culture in that same period of time that the Hanna group uses it in deriving their embryo cultures from those embryo founder cells. So just to put a pin in it for so we can move on. I think I misapprehended the simplicity of the De Silva model, and that's what I kind of conveyed, that it was a technical tour de force from the Hanna group, and the De Silva innovation was that it simplified things. And I stand by that in part, and that these embryo founder cells are a unique cell type. I think that really is a seminal step forward. But I think I really did a disservice to the Hanna group in making it seem perhaps that their elements were more technically complicated, which is not exactly true, and understating the fact that the embryo culture, that roller culture, which was a Hanna innovation, was actually critical to getting the embryo models as far as they did in the De Silva paper. So I just want to acknowledge that. My apologies to the Hanna group for maybe misrelaying the information. And also, just to finish, I want to underscore here that while we're all competing here, I think that this is a testament to, one, that the work moving forward from many different labs and how they're pushing the envelope. Also, that while we are competing here, the Hanna group has been open to sharing this technology with many groups. And in fact, the De Silva work in part is standing on the shoulders of that innovation. So again, my apologies to the Hanna group, and congratulations to you both for an amazing piece of work that's going to resonate for many, forever, pretty much. I think this is really a foundational work that a lot of groups are going to build on. So kudos to you both. Moving on to my current roundup story. This is a bit of a callback to a recent guest on the show, Dr. James Briscoe. And this story is really, I think, epitomizes what I like to call the ODB. That's not the old dirty bastard from the Wu-Tang Clan, but the old school, original developmental biologist approach here, of which James Briscoe and co-author, this is just two authors on this paper, which is another real old school science thing, which I love when you really thin out the author list to underscore that this is kind of, I don't want to say a silo, but this is kind of old school. Very thin author list. Dirk Benzinger, the lead on this. And the nature of the work is also very old school. We're talking about morphogen gradients and the fundamentals of developmental biology. I really recommend you guys go back and listen to that show because it comes through in the conversation that James and his team are really trying to get at the underpinnings, the foundational questions in developmental biology that brought a lot of us into the field before I think embryonic stem cells were ever really a draw in their clinical impact. So this is a story that I think illustrates that approach, and it is about morphogen gradients, which are critical in directing spatial patterning and regulating these developmental programs across tissues, particularly during gastrulation and early organogenesis. And the key is that they're dynamic, right? Because you have all this happening in the context of a growing embryo. So there's a spatial element and there's the temporal element. These developmental programs or clocks are all kind of fixed and intrinsic to the cells. And there's all these complex feedback mechanisms, right? So it's a moving target. And as I said, there's a range of time and length scales that are really specific to each species and the developmental program. So if we're going to try and understand these things, we got to be able to both control, kind of perturb and modulate those morphogen levels and gradients and directions. As well as measure the output, right? To see the cellular response and to do both those things with really fine spatial and temporal precision. And that's, of course, super challenging in living embryos. How do you even get in there? But there have been efforts. So I think a lot of focus has been directed towards these stem cell differentiation models, particularly now as the embryo models are emerging with cells like these embryo founder cells and technology like the roller cultures. We're able to control these inputs with a much finer degree of precision. And there have been models that have been made efforts to control this. There have been like little beads of Sonic Hedgehog. I can remember Studer Group came up with a story many years ago that put this nidus of Sonic Hedgehog that kind of created this radial gradient source and sink kind of approach. And there's also, cell mixing that's been used, but oftentimes it's tough to control, the on-off here. And even when there has been an on-off control with optogenetics, which is, we're going to come back to that because that's what this story also uses. But the approach is one of which, at least, has been generated by my former postdoctoral mentor, Ali Brivanlo and his group. They use optogenetics combined with Cre recombinase to turn on Sonic Hedgehog, which again, this is what's is being used in this story, Sonic Hedgehog, optogenetics. But in that case, they can't turn it off. It's irreversible. So there's a lot of challenges here. How do you turn it on and then also turn it off? Last thing, even when you have a measure of control with these things, it's hard to get a dynamic range that's really tight, right? And relevant physiologically, biologically. You either have something that's a little bit leaky, that even in the context of optogenetics, for example, even when there is no light shine, just the ambient light as the cells come in and out of the incubator, or other reasons you get leakiness, or sometimes they're regulated too tightly, and then you can't get the morphogen or whatever you want expressed up into a range where you have a physiological influence, right? So they went after all of this in this developmental cell paper, which, as I said, just two authors, Dirk Benzinger, James Briscoe, both co-corresponding from the Crick. And what they did here, just starting from the technical standpoint, is they combined they did like multiple layers of control, and that was the key. They had this, what they call this GABBO, which is this GAL4 DNA binding domain, which is linked to this vivid light oxygen voltage domain, which is the optogenetic component, and then a P65 transcriptional activation domain. So those three components create that optogenetic response unit to turn on Sonic Hedgehog. And then here's the key: they went to a next level of negative regulation on that. And I think that's the major innovation here, where they fused that GAVPO complex to a degron domain that was controlled by a small molecule. So only when they add the small molecule do they get this degron and therefore activate and make that GAVPO responsive to light. So it's got these multiple layers of control that they incorporate into a stem cell model. This is in mouse, and in so doing, they are able to generate long-term or long-range, sonic hedgehog gradients that enabled patterning of the neural tube into spatially distinct domains. And here's the key: they had this not only the measure of control, but they fused it to this scarlet fluorescent protein, so they could very specifically see when it was going on and off. And then they could look at downstream gene expression in the context of this very tight control. And here for me was a great takeaway. Sometimes you read this paper and, not for nothing, I don't really care about neural induction, but I took away from here a key point, which I'm incorporating into my cell cultures right now. And that is they found that Sonic Hedgehog has an extracellular half-life below 1.5 hours, around 90 minutes. So right now I'm banging Sonic Hedgehog. I'm having some trouble with my diffs getting the efficiency I want. So one approach I'm taking is I'm trying to increase the interval there where I add in to supplement the Sonic Hedgehog. I had no idea that any of these molecules could act on such a short time scale. And the other key is that when they look at the downstream gene expression, they found that those extend beyond that half-life and that temporal range of the Sonic Hedgehog, which suggests that Sonic Hedgehog morphogen gradient is continually renewed. The patterning is continually re-exerted with the reestablishment and renewal of that morphogen gradient. So hence my turning to the continual supplementation in my cultures. And then finally, they show that it's the acquisition of downstream fate and maintenance of those fates is contingent on not only the concentration of hedgehog, but the duration. So it really underscores how willy-nilly we're getting after these things with our ads. When in biology, you have a constant source that is able to fine-tune development and why you're able to get such a reproducible morphogen output in an embryo, a well-formed embryo most of the time, while we're getting these kind of approximations, at least with the embryo models. But I would say and argue that even in our diffs and EBs, we're really getting these crude amalgams of cells because we have these extrinsic modulators that are not even close to the physiological timing or dose on a spatial scale that is required for proper patterning. So a really cool story for me. I love that ODB element of it, and instructive for me personally, and for everyone else. I think a great framework for building out their own precise morphogen gradients, incorporating into these multidimensional orthogonal engineering approaches to get towards something that really is, a closer approximation of development.
Never thought we'd get a Wu-Tang Clan reference on the podcast, but here we are in August 2025. Yeah, this is a multi-part study. It seems very simple. Obviously, there's only two authors here, including James Briscoe, who we had on the show. But there's a lot into this. First is, of course, the utility of optogenetics, which has become such a mainstay technology, not just in neuroscience, but even in cardiac biology. I remember back in grad school at Stanford, first learning about the amazing work of Carl Dice Roth and also Feng Zhang and developing some of these optogenetic modalities initially for probing neuronal behavior, but then inevitably was adapted by cardiac biologists. Of course, our cell types are also electrophysiologically active. And so I remember in Jobu's lab, we had folks like Oscar Abules, whose work we covered recently. He actually incorporated some of these optogenetic techniques to modulate cardiac functionality by pacing cardiomyocytes with light, which is really, really cool to see back in the day. So that's part of it is the power of optogenetics. And here we are in 2025, and we're still trying to figure out the dynamics of Sonic Hedgehog, which is, I think, pretty wild, pretty exciting. But yeah, I think a reflection of what you were just saying. The systems that we're using in vitro are, of course, artificial, and we're missing this dynamic spatiotemporal control of these morphogen ingredients, such as what happens in the in vivo system. And maybe that's why, going back to your note at the beginning of the roundup, maybe that's why the roller cultures are useful in that way, because maybe you're getting a constant refresh of those morphogens in those developing embryos. I don't know, maybe that's one way to think of it. And one other thing is, of course, the fluorescent reporters. I've used M Scarlet. It's extremely bright, and that really enables the downstream analyses here. But just to get to your earlier existential crisis that you were having of about the time scale of some of these molecules, they actually refer to it in their limitations. It's possible, and they're not able to exclude that Sonic Hedgehog may be cleared on an even faster time scale than 90 minutes, maybe half-life of individual minutes, less than 10 minutes. So, yeah, these are still things that we're trying to get to the bottom of in terms of the time scale of the clearing of these morphage ingredients and how quickly they're refreshed. But these are very, very short scales here.
Yeah, you talk about timescales. It may be 2025, but Arun, Wu-Tang is forever. And you ought to know that coming out of them Wu labs. But yeah, getting back to the science, this is, I agree with you. It's a bombshell on many fronts. Illustrates the tremendous impact. Here we are, more than a decade later. I think downstream of the tremendous innovation from Boyd and Dizeroth of optogenetics. So illustrating the utility, but yeah, getting back to that Sonic Hedgehog thing, everybody loves Wnt, but maybe the linchpin here in controlling things are these things that are ephemeral and it's going to be expensive, Arun, but I am jacking up the hedgehog both in terms of frequency and dose. And I mean, I'll get back to you on whether the Briscoe ODB approach saves my cultures.
You keep me posted on that. I am very curious. Moving on to another study, this time in Cell, DevCell going to Cell here, staying in the Cell Press journals. This is coming from Paul Tissar, who's actually going to be on the show not too long from now. This is focusing on another aspect of early neuronal development, potentially, and adjacent cell types in the brain. We're looking at oligodendrocyte maturation here. And the title of the paper here is Transient Gene Melding Governs the Timing of Oligodendrocyte Maturation. And iPSCs were a mainstay in terms of being the model system that was driving some of these observations. First author here was Kevin Allen, like I said, coming from Paul Tissar's lab. So cell maturation, we talk about this in many contexts, and you name your iPS-derived tissue type, we all have to deal with this in our iPS cultures. For me, I have to deal with it as a cardiac biologist. We got to make our cardiomyocytes one-to-one with adult human cardiomyocytes. We're not there yet. We're getting closer. But here in the context of the central nervous system, you, of course, have to observe that cell maturation as well, not just in your in vitro differentiations, but in vivo during development as well. So the failure of oligodendrocyte maturation, and oligodendrocytes are, of course, these support cells that generate myelin and misregulated in diseases like multiple sclerosis, for example. The failure of the maturation of these oligodendrocytes is indeed linked to diseases like multiple sclerosis, MS. And here, what they're doing in the Tazar lab is diving deep into the transcriptional mechanism that actually governs the timing of the maturation of oligodendrocytes. So, after progenitor cells are differentiating into immature oligodendrocytes, they actually find this single transcription factor, SOX6, one of the canonical SOX family of transcription factors, that was able to really serve as an all-in-one hub for modulating the maturation of oligodendrocytes. So the transcription factor OX6 is able to redistribute from other super enhancers to actually cluster across different gene bodies. And these different gene sites are showing pretty extensive chromatin decondensation, transcription, which turn off during maturation. So SOC6, the presence of SOC6 looks like it's enhancing that immaturity phenotype. And when you suppress SOC6, it deactivates those immaturity loci and actually accelerates the transition to mature myelating oligodendrocytes, which is exactly what you want, especially for potentially a clinical translational context in the disease, in the setting of a disease like MS. Um, Cells that harbor an immature SOC6 gene signature are enriched in MS patient brains. So they actually went back and got the clinical samples, showing that these individuals actually have an immature SOC6 gene signature. And most importantly, well, one of the most important things was they actually did a drug treatment, kind of an antisense oligonucleotide mediated SOC6 knockdown in mice, which was actually able to drive further maturation of oligodendrocytes, which is super cool. So you're going from the in vitro observation in the diffs, identifying SOC6 as this immaturity factor, showing that it's found abundantly in MS patient brains, and then finally wrapping it up with the knockdown, showing that when you knock down immature SOC6 gene signature, you actually get a better maturation phenotype in vivo. This is an amazing study showing that it's really this one critical factor, SOC6, this amazing transcription factor, that's a regulator for oligodendrocyte maturation. And it's not just that, it's potential as a therapeutic target to potentially modulate myelination in diseases such as MS. So this is, I think, an amazing study from Paul Tsar's group. I mean, just a reflection of the amazing work they've been doing over the years. And I'm really excited to chat with Paul in a few weeks.
Yeah, I love this story. And, if the first was ODB approach and this is the NDB approach, of course, that's nonsense. This is all innovative and new science. But this, I think, is emblematic maybe of how stem cells and I mean, I don't know. Paul Tazar has always been so deep on the mechanism, and as you just summarized here, this goes soup to nuts, incorporating these, the original ideas of stem cells, and then taking them to the next level with the molecular biology. So yeah, kind of. I don't want to call it the new, the old. That's kind of dumb, but there's so many moving parts here. And yeah, again, as you summarized, going from the model to showing the disease relevance in human and turning the mouse and the mechanism, and then finally with the ASO, goes really A to Z. And for me, what I'm left with is kind of like the conceptual advance, and it's in the title. I love it when the concept is introduced with a metaphor, in this case, the melting. And the combination of this melting with the retention of a immature identity. Usually think of the activation of maturation. Like it's acquisition, but here it's about shedding the immature phenotype. So just a lot of different ideas here that converge and get me thinking. The best kind of paper for me is to read one where you see the tech, you see the disease relevance, and also it just gets you thinking in a more kind of whole organism context about how the molecular biology might work.
Yeah, this is kind of an old school in that paper because they are taking that mechanistic deep dive and finding SOC6, but it's definitely new school in the technologies that they're using and the really translational element of this towards the end. I mean, that's honestly why I love this. But one thing to note, they're at the translational element at the far side of the paper, their oligodendrocyte work was driving maturation of these oligodendrocytes via knockdown SOC6 in vivo during early postnatal mouse development. And in a disease like MS, which has adult manifestations, I think we still got to figure out if that's feasible, if that's actually a viable solution. Adult versus early postnatal, very different phenotypes there. And of course, mouse versus human as well. But I think a great starting point, if nothing else.
Yeah, and these ASOs are starting to dribble into the clinic too. So I feel like there's going to be a lot more force behind the that class of candidate, which should propel this. So I'm waiting to see where it goes, and we got to talk to Paul about this when he comes on the show. Exciting work. Can't wait. Moving on to some more exciting work here, talking about CAR T here. It's kind of like the singularity with CAR T, where we're going to approach a world where CAR T is used for everything. And we alluded in the past, I think you covered the story, it was years ago, where the CAR Ts were used to target FAP, this fibroblast associated protein in the context of cardiac fibrosis, which was so exciting. And we just haven't heard much about that since, but it's still going. This approach of leveraging CAR T, not just for, cell based disease like cancer, but also other cell derived pathologies, in this case, fibrosis, are still out there. And here's a great example in cell stem cell from Fan Yi's group, who's at the Key Laboratory in Shandong University in China. And this is about kidney fibrosis, right? So kidney fibrosis, it's a hallmark of chronic kidney disease, and there's no anti-fibrotic therapies available for kidney disease. Not really very effective anti-fibrotic therapies, period, although some do exist. But specifically for the kidney, there's nothing that works. And the other part there is that we don't understand with clarity what the origin or what the kind of biology of the scar-forming cells in kidney fibrosis are. So, as I said, there was this idea, or there was this demonstration of CAR Ts targeting fibroblast activation protein, sorry, called associated fibroblast activation protein, that we used to rescue cardiac function in mice, and it was shown that it could reduce cardiac fibrosis. But there were many challenges in the interim. There was increased infiltration of macrophages into the hearts of those mice, disruption of wound healing. You had cachexia, is that how you say that, in mice, anemia. So it seemed like there was like a toxicity associated with those CAR Ts, perhaps not surprising when you think about maybe the promiscuity of this fibroblast activation protein. So there were kind of clinical concerns in moving that to humans. Could be pretty toxic. And also important is that there wasn't really a demonstration that this FAP would be relevant in other fibroblast associated conditions. So myofibroblasts, as you know, Arun, are the major source of extracellular matrix and tissue fibrosis, specifically in the heart. And it's well known that those arise from cardiac fibroblasts in a process that perhaps recapitulates the neonatal or embryonic cardiogenesis. In the kidney, the origin of myofibroblasts is a bit more complex, but recently with these cell atlases of in mouse and human kidneys in the context of health and fibrosis, there have been major strides in understanding what the complement of those cells are. So building on this, the Yi Lab developed this approach where they did a bunch of seq to identify a novel candidate in this mouse model of kidney fibrosis. And what they settled on was platelet-derived growth factor receptor beta as a potential antigen that they could leverage for targeting with CAR T. And I know what you're thinking, Arun. PDGFR beta, holy camole, isn't that on everything, including like all parasites? And yes, the answer is yes. We don't want CAR T vascular attack, but hey, what they showed here was that by either adoptive transfer or using these CD5 lipid nanoparticle LNPs, a liquid nanoparticle mediated like in situ, in vivo generation of CAR T cells that targeted PDGFR beta, they were able to significantly mitigate kidney fibrosis, but also myocardial fibrosis, perivascular fibrosis. You got a 3-for-1 here. And the key being is that they didn't note a lot of toxicity, without notable toxicity. Although I'm sure the devil there is in the details, and this is a mouse model. So we're going to have to go deeper on that. But I'm excited, right? As I said, the CAR T is approaching singularity, where if you are unfortunate enough to be affected with pretty much, well, a whole range of diseases, cancer, fibrosis of multiple organs. There seems to be this one shot CAR T that can address multiple pathologies. And as we talked about, I think it was last episode, the technology with these CAR Ts is getting much more advanced in terms of their persistence, their specificity. So I love this story. As I'm aging and fibrosing across the map, I love this idea of a one-shot approach for multi-organ fibrosis because let's be honest here, when you have chronic kidney disease, cardiovascular fibrosis is very often a sequelae, and there is a lot of interrelationship amongst these fibrotic conditions. So having a silver bullet, although that's an optimistic assessment, as an idea at least, having a one-shot approach that is, call it a cure. Pretty exciting, Arun. What do you think?
Yeah, man. Give me some CAR T, give me some Yamanaka factors, turn my body, take me back in time and put me back in kindergarten, right? That's what we're going for here, I suppose. But PDGFR beta, whoa, whoa. Yeah, that was my big reservation off the bat. But I think they addressed it pretty well and they were pretty straight up with it in the limitations. They're like, yeah, we know PDGFR beta is everywhere. So they got to look at future iterations of the approach and seeing how you can combine these things and target other major surface antigens that are expressed by ECM producing cells. And they list a whole bunch of them, CDH11, a whole ton of them, right? So you got to maximize the efficacy while limiting the side effects, and that's kind of what we're always afraid of is the side effects in these stories, especially with CAR T. And also, there was pretty limited persistence of these things in vivo, and I think that's something we really need to evaluate, especially when it comes to the human therapy side of things. And on that note, they demonstrated that they have the anti-fibrotic effects of these PDGFR beta CAR T targeting T cells uh, on the human pluripotent stem cell derived kidney organoid model that they showed in the study, which is really cool that they actually used an organoid model as their kind of approximation of kidney fibrosis, but it's an organoid model, right? And of course, you have to further demonstrate the safety and efficacy of that in clinical trials ultimately. And this is really, really interesting. Like you said, the potential of targeting multiple organs and fibrosis across tissue types is really promising, but you gotta keep on fleshing this out. So I'm gonna stay tuned here. I'm gonna stay tuned on the Yamanaka factor stuff too.
Yeah, I'm glad you mentioned that they moved it into a human model, because that is critical. I mean, first question is what? Maybe mice don't have PDGFR beta. I mean, just kidding, but important to show in the human. But as you said, when you have the organoid model divorced from the rest of human physiology, which really is the point of your underscoring the necessity for a little bit more preclinical analysis. You're not quite there, but I think there the key showing that this also works in a human paradigm, right? It also works in human cells, which is a key step forward. But yeah, it's gonna be a long road, right? Because I don't think you need to come up with such a high bar justifying the lack of toxicity before you move into phase one with humans, which meaning, larger animal and then primate. I think you need before that. I'm sure that's ongoing, but it's gonna take time, right? Those trials, you have to monitor those animals. I don't know what the timescales that you have to monitor them over, because that is the double-edged sword here. We joke, but wow, think about it. If you could have a CAR T, of course, not when you're healthy, but when you have disease. And bear in mind, I think it's an important consideration that the toxicity just has to be less than the benefit, right? It's significantly less, but there's no treatment for these conditions or no effective treatment. So it's kind of like a Faustian bargain there. But yeah, it is a double-edged sword because the efficacy is predicated on these things living inside you, and their persistence could be a boon in that you have this vigilant surveillance for fibrosis, I guess, like forever young. I mean, think about that. Awesome. But that persistence could also be a liability if it becomes malignant or has any malignant side effect. So yeah, it's going to be a long road to the clinic for this, but conceptually, I love it, and I love that we've circled back to this, the CAR T for fibrosis, because I thought maybe it was not dead in the water, but had some insurmountable challenges. It's good to know that people are still cracking on it.
Yeah, it's still alive and kicking and extending to multiple organ types and potentially an all in one shot, which is like super, super cool to see. Um, one thing we found out when we were at ISCR in Hong Kong is that China is really taking a leadership role in some of these really cutting edge clinical approaches and translationally oriented stem cell approaches. And that's true in this story that you just covered, and also in the last story of the roundup that I'm going to talk about here today, which is talking about one of my favorite topics, as we know here on the podcast, and that's xenotransplantation. This is a Nature Medicine story coming from the last author of Xin Shu, but also there's just a ton of folks on this study, as you might expect, for such a clinically and translationally oriented study. This is, like I said, coming from Guangzhou Medical University in Guangzhou, China. Um, First author here is Shansheng He. Pig to human lung xenotransplantation into a brain dead recipient. And I I'm pretty sure this is the first time this has been done. Yeah, I mean, right there, genetically engineered pig lungs have not previously been transplanted into humans. So there you go. I mean, we've been talking about other tissues in this context, the heart, kidney, but lung is a leap further than that for sure. And so there's a lot of questions here that we have to answer regarding the human immune response in the context of a xenotransplanted lung. And I guess, for just a couple seconds, I can talk about why this is important, even though we've talked about this a million times. Of course, there's a huge shortage of human organs for transplant. And the thought is, why not make immunocompatible pig organs? Because we already mass produce pigs anyways, right? Pigs have similar sizes in terms of their organs, in terms of compatibility for transplanting into humans. So, and this, like I said, this has already been done in some capacity for the heart, for the kidney, and extending this to the lung. But immune rejection, that's always the number one thing when you have to think about when it comes to transplanting these immunocompatible pig organs. So here they reported a case of pig to human lung xenotransplantation, where they had a lung from a 6 gene edited pig. Presumably, the edits are, of course, related to immune modulation. And this lung was transplanted into a 39-year-old brain dead, a male human recipient after a brain hemorrhage. And I didn't dive too deep into this, but I think that was the appropriate situation for such a cutting edge approach like this. Initially at least, before progressing into other individuals. So what they found is that the lung xenograft that had these immune edits was able to maintain viability, functionality over the course of 216 hours. So that's like 9 days, just about, of the monitoring period without any signs of the hyperacute rejection or infection, which is great. That's the first step. Getting beyond that immediate hyperacute rejection where the body's just like, whoa, what are you putting into me? But then beyond that, they did find severe edema resembling, primary graft dysfunction at, well, this is actually earlier, about 24 hours of transplantation. They did find some severe edema, potentially due to ischemia reperfusion injury, which is always an issue with transplants. And then they did eventually see the antibody mediated rejection appear around day 6 or so, partial recovery by day 9. They're alluding to it, but the other thing they mentioned here, there was extremely heavy immunosuppression here with like everything that you can think about, rituximab, like all the stuff that you'd expect, tacrolimus, bunch of steroids. And yeah, so eventually, there was the rejection, right? But it's a proof of concept, feasibility, showing that you can have feasible pig to human lung xenotransplantation with getting beyond the hyperacute immune rejection. Um, but a lot of challenges still remain here. And in general, they do remain in the xeno field, even though we are an order of magnitude further than where we were 10 years ago. Um, I've gone on the record and said that I still think this is the ultimate solution to the to the organ shortage crisis is Xeno, as opposed to, and cover your ears, everybody in the stem cell field, as opposed to cell therapies, for everything except for the brain, because I still don't want a pig brain. But I think for large organs like the heart, the lung, the kidney, I still think Xeno is the way to go.
Partner, my wife will tell you that a pig brain for me would be an improvement.
I was waiting for the joke to show up. I knew it was coming.
Couldn't help myself. But this is, I don't know, I'm both hot and cold on this because one, like you just finished with, so we have heart, kidney, liver, and now lung. That's all the vital organs that maybe not all of them, maybe I'm missing something, but it's a lot of them, right? Where people are like, how can we do this with cells? There may be some insurmountable challenges. So, wow, that it seems like we closed the loop on the big four there, not including the brain. And the other side of it is I'm not really, I feel like this is a negative result, kind of, because it's not. I'll put it this way: I wasn't like, oh wow, green, brighter days ahead. Like, let me go start smoking because it doesn't seem like this worked very well. I'm not about to start smoking. It's not like I started drinking when I saw a liver one, but I will say that I was more optimistic about the progress in those, whereas this one I was like, oh damn, it doesn't look like it worked. But or it looks like we've got a lot more challenges here. Why didn't we have those same challenges with these other organs? I guess is the question I'm left with. And one question I have is there some difference in the input? Like these, Revivicor, that United Therapeutics based pig, is that different from these other pigs? Like eGenesis has another approach. Is this its own like bespoke modification, or is there like a central? I don't know. I guess you could probably find those details in the paper. But whether or not that's the case, I think we need to try the lung from the same pig that where it's worked with the kidney. I think we need to try the lung in that from that pig before I make a judgment on this, because I'll be bummed if the kidney works well or any organ works well and then the lung is just has fundamental obstacles. Not super bummed, right? Because hey, you can't have it all. But more, I would be haunted by the idea that okay, what you're seeing in the lung when you look at this whole organ transplant of a hypoimmune pig, what you see in the lung that you don't see in the kidney, it's just a matter of time before you see it in the kidney. It's hard for me to believe that there's such a fundamental difference. Maybe point to the vascular density or something, which could be. But I don't know. I feel like if we can't get as good a result with the lung as we have with the other organs, I'm a little bit spooked.
Yeah, and that's exactly what they pointed at, was the vascular density for the lung as to why this may not have worked as well. But then again, I mean, the kidney is also pretty heavily vascularized, as is the liver and to some extent the heart. So I I think you're right. And maybe they're doing this on the Revivicor and United Therapeutics pigs. They're gonna try the lung. And I don't know how different those are versus these versus the eGenesis ones from George Church and all of that, right? But I think getting beyond the hyperacute rejection is still a plus, because
For sure.
otherwise this thing would have been non-viable in hours as opposed to days, and so that's still great progress. But you're right. I think a lot of work still remaining to be done here.
Cool though. So cool. I mean, what a crazy show we had today. We got the CAR T taking care of all that, and we got the whole organs. Wow. I mean, I'm not going to start smoking, but I don't feel so bad about my choices in college nowadays. Uh, Moving on to our guest, we got a great one. I'm going to have a nice chat with Alice in just a sec. Before we get there, though, quick message from STEMCELL Technologies. Take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance, is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. All right, everybody. Today from Arun's neck of the woods, we have Dr. Alice Soragni, who is associate professor at the University of California, Los Angeles, UCLA. The Soragni lab investigates how protein aggregation affects cancer development and progression with a special focus on P53 aggregation. We're really excited to get into this with Alice. They also focus on developing clinically relevant organoid models for cancer biology. That's a little bit more in our wheelhouse. We love the organoids. Also working on drug discovery and development applications for those cancer organoids. In particular, they establish tumor organoids from surgical specimens, which are patient-derived, and perform high-throughput drug screenings to individualize cancer therapy to each patient, which is the future we all know, personalized medicine. Alice, bringing it to us and bringing the conversation to you. Thank you so much for joining us today.
Thank you for having me. Very much looking forward to our chat.
Yeah, great to have you on the show, my dear neighbor down at UCLA. Dale gave a nice overview of kind of what your lab is working on when it comes to tumor organoids, protein aggregation, but we're going to put you on the spot. So why don't you kind of summarize in your own words what your lab is doing and really kind of what excites you most about your current research?
Yeah, so I mean, I think probably the most exciting part of what we are doing is the focus on developing patient-derived models of cancer and of malignancies in general. So, we particularly focused in the field of rare tumors and rare cancers, and so that's a place where models are scarce, and sometimes there is no model at all, and that truly makes finding new effective therapies, testing drugs, and so on and so forth, virtually impossible, right? Some of the diseases we work with, there isn't even a single cell line. There is absolutely nothing. We also have a very strong focus on not only going after your, quote unquote, conventional carcinoma type cancers, but also things that are, non-epithelial tumors, benign tumors, slow growing tumors. So those are all things that are so challenging to, to make a cell line out of, to grow in a mouse. And so you are in this kind of, model desert, as I like to call it. There is nothing. And how do we come up with ways to, improve outcomes for patients, find new therapeutic avenues, learn more about the biology of these tumors, which, sometimes we know so little of. And so I really think organoids are, it's such a sweet spot. They can really help us model these diseases, which we know very little of, and come up with new therapies and new ways to look at them. And so that is probably my the part that is more exciting, particularly again when you focus on these very, very rare, some of the things we work on, there is maybe 20 cases a year in the US. Some, there has been maybe 50 cases over the last 70 years. I mean, these are rare things, right? That no one has looked at before. So I guess makes me feel a little bit like an explorer as we go and dig into these and look at it in a way that no one has been able to do before. And so I find that very exciting, I guess.
Yeah, I love the analogy of the explorer, right? Because there's that element of every one of these cancers that are so rarely studied. You're kind of on new ground and unraveling truly novel biological mechanisms, malignant in this case. But that's really the whole promise of the personalized medicine. We're going to expand on this in just a minute, but I love that idea of going outside the traditional pharma paradigm, right? Where you need a critical mass of patients in order for the economy to make sense. But that's the whole promise of personalized medicine. And I think you're a great example of it, going all across the board, all these tumors. As I said, we're going to get into that. But first, I want to focus on kind of a linchpin, I guess, of your approach, which is fundamentally structure, right? Your doctorate was in physical chemistry. You spent
Oh,
years
yes.
doing
That
NMR.
structure.
I can
Yes, it
remember.
was.
Oh boy, and that is, I mean, I can remember from my graduate school days, this, solving structure. I mean, that was 100 years ago. I'm sure it's better now, but still, we're talking about really exacting, work that focuses on the minutia, highly detailed, very chemistry associated, but really focus on 3D organization at the molecular level. That's the bottom line. And you're now applying a skill set to unravel the role of malignant protein aggregation and cancer progression. I can remember when I hear about protein aggregation in the malignant context, it's usually, well, it's not, it's a few things, but I can remember years ago, there was like the prion thing with the mad cow disease, Creutzfeldt-Jakob disease, that kind of introduced the wide world to prion-based diseases. But then, of course, the more, run-of-the-mill, call them, diseases, neurodegenerative conditions, a lot of them like ALS, Parkinson's, Alzheimer's, Huntington's. But I hadn't really been aware of the role of protein aggregation in cancer until I looked into your work. Can you walk us through the mechanisms of action there? How does protein aggregation lead to cancer progression and or, other kind of mortality, morbidity?
Yeah, and I think it's it's sometimes interesting to go back to the history, right? Yes, I do have a PhD in physical chemistry. I come from a very physical chemistry, structural biology background, but that's what got me into protein aggregation, right? And so it was really I was coming at it from the neurodegeneration angle, just like you said, right? Like we know protein aggregation is so deeply entangled with the development of those neurodegenerative conditions. And so that is what I was originally studying, right? Going after Alzheimer's and Parkinson's and some of these diseases where the brain is, ends up being riddled with protein aggregates. And, I think we're not going to touch probably the Alzheimer's side of things, which is a little bit controversial as to what's happening and what's the role of what and what's, but, generally speaking, I think we can say that protein aggregation in the brain is not a good thing to happen. And it's been very challenging to come up, I think, with way to therapeutically approach that in a way that changes disease progression and is meaningful and so on. So it's from my perspective, it was also like, it's so hard to try to keep these flimsy, old neurons we have alive when you have something like protein aggregation that makes them, kind of worsen. But protein aggregation, it's just a very general phenomenon. And so what happened is when I started my postdoc, and I was coming actually to UCLA to do my postdoc with David Eisenberg to get structural biologist for protein aggregation in neurodegeneration, there was this like very exciting paper coming out showing that P53, so master tumor suppressor, guardian of the genome, very important protein to keep tumors from happening, can actually aggregate in cancer cells. And so that was, a very exciting thing to go after and really understand, why is P53 aggregating in these tumor cells? What's happening there? And so, that was kind of the into cancer, I guess, and cancer biology. So we started looking at P53, and what happens when it aggregates. So, typically in tumor cells, majority of them, any tumor cells pretty much has to get rid of P53 one way or another to turn into a tumor cell. And so some of them do this through mutation. Once the protein mutates, it's very prone to aggregate, and that kind of takes the protein away from its tumor suppressive functions really well. And so for a cancer cell, it's really advantageous of getting rid of P53 that way, and any other way really, but that is a very good way to just kind of take it away and be happier, go on and develop as a tumor. And so, in my postdoc, because that was the approach of, the Eisenberg lab, we looked at developing inhibitors of the aggregation process with the whole idea that if you can stop p53 from aggregating in these cells, you can actually make it functional again. And once it's functional, its role is to, get rid of tumors, right? And so that was the whole idea and how we started. And so we really see p53 aggregation as a way for a cancer cell to increase its fitness and propagate, further. And so the idea is, can we stop that? So that's kind of how it came about, yeah. And we're continuing a little bit of that work still now, although I'll say that our rare cancer tumor organoid angle has probably taken over the majority of my lab, but we're continuing that. We're looking at p53 aggregation in early, tumor development to understand how early is that, misfolding and aggregation happening. And is it something we can target, for example, tumor prevention in certain settings and so on and so forth. But also looking at aggregation more broadly, like cancer cells are very capable of buffering a lot of, stressful conditions and survive them very successfully. And so we're really interested in seeing whether changes in protein aggregation, depending on the condition that the tumor cell undergoes, could be a way for the tumor cell to, again, increase its fitness, right? And so those are the things we're still looking at.
Yeah, so you mentioned how your lab is really shifting focus and focusing very heavily on these tumor organoid models. So let's actually talk a little bit about that. I wanted to highlight one of your recent efforts. It was a paper I think that just came out in Cell Reports Methods, if I'm right. It's about this patient-derived cutaneous neurofibroma organoids for high-throughput drug screening. And I mean, this is super impressive. I remember you actually talking about this at our recent organoid symposium that we had at Cedars-Sinai, for which you were a speaker. So thanks for that. And also, there's this really cool mini ring platform that lets you test treatments quickly and at scale. So talk more about this study, this platform, how it's kind of expanded your ability to study these rare tumors, and talk more about it.
And it's really kind of, it all came together during my postdoc, truly, because as we were developing this inhibitor of p53 aggregation, we wanted to test that on real, quote unquote, real tumors, right? And so that's how I started establishing patient-derived tumor organoids for ovarian cancer at that time. And then when I started my lab, suddenly I had this very specific expertise in developing patient-derived tumor organoids that, was becoming so exciting. The field was really kind of opening up to this idea of organoid technologies in the cancer realm as well. But what we realized very quickly is that I wanted to know more about this tumor. I wanted to poke and prod them in as many conditions as possible. But it was very challenging to perform drug screening in 3D. It remains very challenging. And so there was a very technical issue at the beginning, which I felt like we need to address right there and then. And so if you want to test a lot of conditions in these organoids, how do we make 3D, drug screening simple? And that's how the mini ring platform came about. We really decided that a lot of the issues we have are due to the way in which we grow things in 3D. Just because they make, using liquid handlers and automation very complex, we like to put things at the center of the well. And that is not intrinsically wrong. It just makes your life a nightmare when you're trying to go into automation and high throughput drug screening. And so that's how we came about, with this idea of just changing the geometry. If we think that we can have an easier life and perform more robust drug screening by having an empty center of the well, let's just develop organoids at the periphery. And so instead of putting our, say, matrigel or whichever matrix you want to use, at the center, we do these rings at the rim of wells. And that is really, as simple as that may be, it's been truly transformative because it has allowed us to do this high throughput drug screening and, not just do them, but produce robust data, right? Data that is solid, that you can have confidence in, and that you can use, for example, to guide therapy eventually, right? So that was the important part of it. And as I said, we have applied that to a lot of different diseases, but cutaneous neurofibroma is near and dear to my heart because I don't know if you guys are familiar with this type of disease, but this is a kind of a tumor that happens on the skin or just under the skin of patients with NF1 syndrome. So these are patients that have a mutation, in the germline on this tumor suppressor called NF1. And once you have, a copy that is altered, you are prone to developing a number of tumors from all the way from benign to highly malignant. And these very, in a way, overlooked symptoms are these cutaneous tumors. And, they're kind of bumps under the skin, but the problem is that some patients develop a few, and some patients develop hundreds, and some patients develop thousands. And these is, entirely benign tumors. These are never going to kill a patient, right? And so, from a medical perspective, it's like, well, that's not, necessarily the biggest issue, right? However, they affect the quality of life of these patients tremendously. And so, and they're also, they cause itching, there is all sorts of issues that go with them. It's not, it's not an easy manifestation of the disease. Is not, and there is truly no systemic therapy just yet for that. So these patients can get surgeries, but again, there is only so much surgery you can do. If you have 1,000 lesions on your body, that's a lot of, tumors. And you can get scarring from that. If it's not completely removed, they can potentially regrow. And so, again, it's a very, in my mind, important condition to address for patients with NF1 syndrome. But then, and then there is the tumors per se. These tumors are hard to model. They are not just made of one cell type. So the tumor, initiating population is this Schwann cell type, tumor-like cells, which has a double hit in the NF1 gene, but half of the tumor is fibroblast, and then there is an immune component that's really, kind of critical to that. And so what we wanted to do going into this is developing an organoid model that allows us to keep all of these different cell populations, right? Because that's really the key. You're not just trying to take a pure population of, tumor-like Schwann cells, because that's not what these tumors are. That's just one component. And so, it's part of why I'm so excited about tumor organoids in the way we do it, which, just for full disclaimer, is unpassaged. So it is passage zero. Short-term culture is that you can keep all these different cell populations there alive and happy. And then you have a platform, right, which you can use to test therapies, to test combination therapies, as well as to learn more about the biology of these.
Yeah, I mean, I should say NF is important to me too. My sister has NF, and she doesn't have, the lesions have never manifested for her in her entire life.
That's fantastic.
Yes.
And that's the thing with this disease, right? Because NF1 is it's a huge gene and can be mutated in so many different ways. We actually do not have just yet a very comprehensive way to study this phenotype genotype kind of relationships, right? So we actually know that there is a subpopulation of patients with NF1, and I'm so glad to hear your sister is one of those, where they have no lesion, but then the next patient has 1,000. And so I think the beauty here is that the organoids give you a cheap and quick way to sample as many patients as possible and really try to understand with all these different genotypes, right, what happens there. So
yeah, and I mean that's the thing. Even though she's lucky and knows it, but she always has agonized over, well, is it ever going to matter? And that's because it hasn't. Is it going to? And I think it reflects how little we know about that. And many of these, I don't know, call them orphan, but yeah, call them orphan diseases, NF, which affects a lot of people. But there's like you said, there's such a broad spectrum, and we don't understand it. So it kind of feels like an orphan disease. It kind of feels patient specific. So yeah, I can see how that can be very, I mean, empowering for patients, but also for you to try and, delve into the phenotype genotype alignment there, as you just said, and also reflective of your tremendous productivity. I mean, you talk about NF because you've gone deep on that, but you've been doing this across the board with all these relatively rare cancers, unlocking the maladaptive mechanisms that drive disease. This recent paper you had in AMBO is another great example where you kind of go different. In the mini ring thing, it was a kind of engineering platform, but here's, I guess, call it traditional, even though this is new science at this point, but you went from a model to the screening approach to a therapeutic candidate. And that success, as well as I think similar progress across the field, really underscores the clinical potential of these personalized cancer therapies. But I'm curious, how do you envision the treatment being deployed? Like, what are like the major, you talk about, of course, is it an IND for every patient? That doesn't really seem practical, but like, what's the paradigm in which you mobilize these treatments? What are the challenges? And like, what would you say is maybe the best use case? I don't know, maybe it's NF, but if not, what do you think is the best use case for personalized therapy to really underscore and make the potential undeniable so we can fast track these types of approaches?
And I mean, I think there is so many good questions in this question, I mean, there are so many current issues and some of the, I guess, regulatory challenges are still kind of, TBD. The field is changing very rapidly. The regulation and deregulation and how all of that is going to evolve, that's a tough topic. But kind of starting from the point of how do we start, right? So say we have this platform, we can screen different drugs, we can find what potentially what works best for each and every patient. What is the best way then to go about that? And I think in my mind, the way in which we wanted to, I guess, start was maybe an easier case and a case where we had enough data to truly be confident that we have a predictive platform at our hand, at our disposal. And so what we did is actually on the tail of the STEMCELL paper we published last year that's focused on sarcoma. So tumors of bone, cartilage, connective tissue, soft tissue, and so non-epithelial tumors, right? That's a family of rare and ultra-rare cancers, which includes osteosarcoma. Osteosarcoma is a bone tumor. It's rare, but it's actually one of the most common tumors in children. And it's a tumor type for which there has been a lot of research, and there is, a very well-detailed kind of standard of care. And so you would think, okay, well, then what do you have to do there with organoids? However, we actually started this year our first clinical trial in osteosarcoma with organoids called PRIMOS trial. And this clinical trial we opened at UCLA is a 2-arm trial where we are really looking at a child coming into the hospital with a potential osteosarcoma diagnosis. And so what happens there is this child will get a biopsy, and then we'll go on a standard of care neoadjuvant therapy, which is a 3-drug regimen that every single patient is going to get, right? So it's a very standard procedure there. But what we found in the paper is that we can take a piece of that biopsy, and then we can develop tumor organoids, and then we can test that standard neoadjuvant therapy. And what we see is that we can, within a week, we have results in a week, right? We can tell how these organoids do, and that matches really well with what happens to the patient. So patient goes on neoadjuvant, a few weeks later, they get surgery. And then when we remove that tumor, that is the time where the pathologist goes in and they look at, did the tumor respond or not? And they do that looking at necrosis. And we know very, very well in osteosarcoma that the amount of response you see in that tumor after neoadjuvant therapy is actually a great indicator of long-term outcome. And so a child that comes in, goes on neoadjuvant, tumor is fantastically necrotic, beautiful response. That child is very likely to do really well, be effectively cured of disease, right? But 3 to 4 children out of 10 do not respond. do not respond well enough, and there is no way for us to tell, right? Every child does the same thing, and then it's like, oops, that didn't work. But in the meantime, the tumor has seen all this therapy, right? And changes and evolves as tumors do. And so the idea is, can we use the organoids to tell in advance which patient is not going to respond to neoadjuvant therapy? And so, again, the first arm is just that. We want to answer this very simple question: is this a good prognostic tool to tell a priori who can respond or not? And so, again, I understand we're starting simple, but the idea is, if we can unlock these small things and prove that it's a prognostic platform on a very, established FDA-approved type of therapy, that's the stepping stone to the next level, right? And so that's the first part. And obviously, it's not like we want to change standard of care tomorrow, but if an oncologist has the information ahead of time that, hey, this is what we're going to do. Oh, wait, I know it doesn't work at all, so maybe we should do something else. You know what I mean? And so that's the simple idea there. And then our second arm is looking at patients with, that are recurrent, metastatic, and so past first-line therapy. Now, what is very interesting, I think, in osteosarcoma is that, contrary to many other rare tumors, there is a lot of FDA-approved, NCCN recommended regimens for second-line therapy. About a dozen. There are a dozen there are 12 options or so that an oncologist can pick and choose from. Now, you have a patient and you have 10 to 12 options. How do you pick? You have no data. You just got to pick, right? And so, to me, it is just, this is such a crazy situation, right? And so what we want to do, the way in which we want to deploy this platform is, can we actually help the oncologist make that decision? Can we make it data-driven, right? Can we say, okay, we tested these 10 things, and these 3 are the ones that work better. Just so maybe you're not picking amongst 10, you're picking amongst 1, 2, 3 options that kind of edge your bet, right? And so, again, it's a very simple kind of de-risk type of situation where you are starting on a background of approved therapies considered safe, but just too many options, right? And so, again, I think for us, that is the starting point. Obviously, there is all of the possibility to do drug repurposing and drug kind of repositioning. And so we can go and screen broadly, right? And find something that's maybe not even a cancer therapy that works. Like, there are all these different options, but then, just as you said, there are regulatory challenges. There are some issues on how you're going to do that. And so, again, we are starting from the simpler questions and kind of settings and building up from there.
Wow. That's a great example. I mean, it's all of it, right? Because first, there's that low-hanging fruit where, as you said, we can glean the information and apply it with, drugs that are already approved. Thanks for sharing that. I want to shift gears dramatically here to, to a subject that I know almost nothing about. Both of us pale in comparison, I'm presuming, to Arun, who is the world expert in the social media dissemination of scientific knowledge.
Not true. Not true.
But I will say, I mean, he was early to recognize the utility of social media for scientific discourse, although maybe he's a little bit more cynical about it now. I don't know. We'll have to ask him. But so were you. And you
was I.
Yeah, way back. I mean, this is like
way back,
a generation in tech terms. It was like Chat GPT versions at 1.0. In 2019, you wrote this piece that elaborated on the value of Twitter. Oh boy, it was still called Twitter back then. That shows you how dated it is. Yeah. And also the challenges. Really, I read the piece. I thought it was great. I learned something, but that's not saying much. The bar is very low. I know nothing. But you expanded on the challenges and benefits of navigating that medium, and a lot has changed, as we said, in the intervening years. But I got to put it to you now, 6 years later, have your sentiments also shifted with the winds? And whether or not, what role do you think social media plays in science, your science, just all science these days?
No, that's a great question. One is that is so near and dear to my heart. And for 1 second, I think if we go back all the way back to 2016, 17, 18, 19, the good times of Twitter, where there was a project, there was something that was, it was such a force in science. And I think, people that tell you that's not true, that never existed, they're just lying, because the reality is that I think Twitter was truly transformative in science and science communication. And it did something that was not achievable before, which is give people like, me, right? Someone who started small with very few resources in not the best known place, to have a voice and go out there and advertise my science and let people know about what I do. It taught me so much. There was so much just, the opportunity to see papers I would never be looking at otherwise, right? The opportunity to have conversation directly with the authors, with the other scientists. And this is true at all levels, right? Like trainees can ask questions, like anyone could participate to that discourse. It was just outstanding. It was a beautiful community. And me together with Annie Barmaitra, it was at MD Anderson back then, we wrote that piece because it felt very timely. The networking and the opportunities, and personally, I have met so many of my collaborators, so many of my friends through that. I've written papers that would not existed. I've started collaborations that would not existed. I've gotten grants that would not exist if it wasn't for Twitter, right? And those connections up there. And so it was sweet, it was a great time. I think it was a time really that gave a voice in science to people which are not part of the usual, old networks necessarily. And some things were very endearing to me. It's like no matter where I go, where I'm going for a conference or for a talk, there is always someone that knows me through Twitter, right? I never felt less alone. It's like, I don't care where I am. I know there is someone, you know what I mean? And it was just so, yeah, I think those were very, very beautiful times back then. Yeah, and I think to me, the funny thing is, we wrote that thing and then we actually didn't know that Nature Review Cancer made the cover out of it. So we have this beautiful cover with little birdies flying around. And then it all came crashing down. That's the only cover I've ever gotten in my whole career. And now it's linked to Twitter. And now that happened. So yeah, so personally, I logged my profile on Twitter. I didn't delete it yet. Many scientists did. And I think that's probably, that is more of akin to my type of response. Some people are still there. But to me, just the sheer degree of misinformation and just mishandling of the platform and the way things come out or are associated with, that's not my neck of the woods anymore. I don't think that's productive. I am again, you will always find a scientist that says, I'm still there, I'm fighting, all the misinformation. Someone has to do that. And that may be true and good for you if you're doing it, that I don't see that as productive or a productive use of my time. And, I think it left a vacuum. It truly, truly did. There are new things now, which I'm fairly excited about. I think Blue Sky is a good alternative. It's a good community that's built in there. Lots of the same folks that moved over. It's still a little bit less volume if you wish, but that's something. And then personally, I started doing something which I never thought I would do, which is use LinkedIn more. Which is like, a little bit different, right? But another way, to communicate your science and talk to folks. Not maybe as dynamic as a platform or as easy to have back and forth, like in comments and chats, but something that's happening. But what I strongly agree with Arun probably is that you got a network, you got to get, your science out there. You got to find those people to work with. No one is an island. for me, in the rare disease setting, that's paramount, right? Like, if there are 10 patients in the US, I need to work with all the people that see those 10 patients, right? Like, you have to work together. You have to build this community. And it's so important for trainees as well. It's important to have those conversations. And so yeah, I guess I'm mostly retracted to Blue Sky and LinkedIn right now. But those golden times, miss them every day.
Oh, the good old days, right?
Ah, the good old days.
Yeah. Yeah, I think in prime Twitter days, it used to be this, amazing community to just openly have scientific discourse and make connections. I totally agree with you. I think that was a huge advantage of just being able to network. And certain folks really took it to the next level because, they used it to get speaking opportunities, to get collaborations, all this.
Absolutely.
Like I
So
was the
did
same
I.
way.
Yeah.
But you mentioned Blue Skies as, nice alternative. And I think a lot of folks have shifted there as well, which is great. It's good to have other options. And yeah, you're totally right. LinkedIn has somehow emerged as this option as well to
Yeah.
mix
And
results.
I mean,
Yeah.
ultimately, when something as big as science Twitter was implodes, it leaves such a vacuum, and the risk of fragmenting across 1,000 options is very real. Certainly happened. I signed up for some platform. I don't even remember their names anymore. You know what I mean? It's like people were really craving and trying to find that alternative, and trial and error, lots of things that did not pan out. And still feels like we are trying to build momentum on Blue Sky. And I say, it's like even myself, right? It's like it's something I enjoy doing, but when you are a scientist and it is still your career, it's not like you're going on Twitter and just, shooting random things. Like, you're trying to put some effort into that communication. Um, so it's a job in the job. Sure, you get a lot of advantages out of it, but you got to work at it. And I guess we're scientists, content creators to some extent, right? And I find in myself having a harder time, getting back into the rhythm of doing that more regularly, which I did at the beginning. But yes, the hiatus was long, and I don't know. It's different, but I'm trying. I think it's so important even now. It's truly important, and also I feel like there is a lot of things we need to speak out about now, and the more publicly you do, of course, within your own comfort level, the better. But it's a tough time for science, and so we got to be there. We got to be prominent and talk about it.
Yeah, absolutely. And I think, you're also at a place like UCLA, which is, of course, a world leader in the work that we do and is able to showcase some of these incredible cutting edge advantages, cutting edge technologies that are emerging in our field, because obviously UCLA has an amazing science communication department and communications department in general that's able to disseminate the work that you're doing. So, perhaps the social media side of your enterprise complements what UCLA is already doing, which is great. I mean, I think a lot of places around the world don't have the name recognition that UCLA does, unfortunately. But that is certainly an advantage of being there. And
I mean, yes and no. I think you're right when you say it does complement, but I've always, particularly when I started, right? And again, those were good times on Twitter, but I feel like that is what set the bar for me. That is where all the effort went. And then I kind of feel like sometimes the communication kind of folks, which are great here, is like, because I had a public persona out there that people would recognize, that gave me more opportunities to work with them, right? Because when you are at a big place and there is a lot of folks doing science, it's not like you can have 1,000 science news going out of your communication department every day. And so again, I think you got to edge yourself out first, probably to get, that's another opportunity you get. Particularly again, if you are not the one, I'm not the HH in my lab here. You know what I mean? It's like you just have to create those opportunities. And so that is another space where you start, with your very democratizing your social media of choice, work, and you build on that.
Yeah, so I mean, let's talk about UCLA just for a little bit, since, like I said, you're my neighbor, right? UCLA, this amazing, academic institute. It's renowned for both stem cell biology, cancer biology. Actually, a new director in the Stem Cell Institute, and Tom Rando, who I actually know pretty well from my days at Stanford. I mean, we've had like we were talking about before the show, we've had a ton of folks from UCLA on the show, including Ming Xiao Gu, who recently relocated from Cincinnati Children's, Aparna Burduri, who's an expert on all things brain organoids. So talk about UCLA. Obviously, things are weird right now across the country when it comes to federal funding. And, I didn't want to get too deep into it, but that is a thing that's happening right now. So talk about
Yeah.
UCLA. What do you love about it, in spite of all the chaos that's happening everywhere right
Yeah.
now?
Challenging times, not gonna lie there. But I think the reason why I have been here a long time, right? I came from my postdoc and I never left. At least yet. And it's just a fantastic place. It's very collaborative. I think people here really focus on working together. There has never been a time where I sent an email to some random person I think I would want to work with, or I think could help in any way, where I didn't get a response, an enthusiastic one at that. People really are, terrific here. My work is very translational, so I depend so much on interacting with physicians at very different levels, and that's not true universally, but our physicians here are absolutely fantastic and also incredibly collaborative. They may not do the science themselves, but they're interested in working with people that do, and those partnerships, are fantastic. So it's been my favorite place for a long time. It's a great place to work at. I think it's a fantastic place to train. We have outstanding students. So those are all great things. And obviously, times are incredibly challenging. Of course, I think that's a universal truth in the US, but at UCLA, particularly over the past few weeks. And I think everyone is kind of on edge, waiting to see what's going to happen.
Hmm. Yes, times are tough, and it doesn't matter whether you're a big institute or small. The outlook seems bleak, although I'm hopeful that it's just a tantrum. But
I don't know. That's a long tantrum. How long can you keep going? Yeah, no. I mean, times are changing. I think realistic. I appreciate the optimism though.
mm.
And a lot of colleagues I talk to do maintain the optimism, and I'm very jealous. My optimism is all used up. And I think what's left now, it's the reality that the science is changing. The landscape is changing in a way that is dramatic and not something we can snap back, even if tomorrow all the decisions are changed. You know what I mean? And so it's scary times, I guess. It's science will continue. I don't think there is anyone who's going to be able to stop science, but we will have less of it, which I think is a problem. And the people that will be able to weather these and survive, you know, is not everybody, so that's a problem. And also, I'm very kind of keen in seeing who's gonna survive, right? Like, who are we gonna selecting for when the times are tough and the resources are scarce, and you have to be creative and go out there and do something different. The beauty of scientists is that we are this community of very different, diverse people. And so who's gonna make it out? I don't know. So
Yeah, it's grim. It's grim. But my thinking there is, first of all, science, everything is all about the future, right? And I think we got to focus on the trainees. And while it is a grim prospect and there is going to be a ton of collateral damage, I can recall 20 years ago with the assault on stem cell work, which was very reasoned. It wasn't as knee-jerk as this or ulterior vested interest as this, but I will say that there was an energy of innovation and promise surrounding it that made it still attractive to trainees. And I think what's great about your work and this focus on, orphan diseases and rare cancers is that it's so exciting, and it paints a picture of our future where we are focused on these individual cases and we're innovating toward that end. And so it is still exciting to be in
It is,
science.
yeah.
And while
Yeah,
there
no,
will be
yeah.
collateral damage, like you said, out the other side, a lot of great science, well, maybe smaller, maybe impacted, but a lot of great science is going to come back on the back end. And that is going to be driven by this next generation of trainees who are looking up to you. So thank you so much for sharing all your work and your thoughts. I love how we went deep into Twitter because I'm such a naif when it comes to it. But I really feel like my knowledge has increased 1,000%. But more than that, your science, and I really appreciate you sharing that with us and the listeners to the show. Before we let you off the hook, here we have a couple peripheral questions here for you. One is, if you were not a scientist, what do you think you'd be doing with your life?
Okay, that is, now that you asked me that question, I realize that I don't actually have a good answer, because my answer is I will always just be a scientist. I will always just be a researcher of some sort. If when I guess I could be a different type of researcher, that I could that as far as I could get. I had a deep interest, in ancient history and archaeology and paleontology growing up. So I could be a different type of researcher. But to me, I feel like I have to be in a place where you are using your curiosity and you are exploring things and you are generating new knowledge in some way, shape, or form. And I'm sure there are a million other fantastic jobs out there that people could do. But again, I'm very jealous, I wish I could say, oh, you know what, if the world is burning around us, there is no money for science anymore. Tomorrow, I'll go and open an ice cream shop in downtown Los Angeles. I'm just not that person. I feel like being a researcher and this kind of knowledge voyage I'm in, it's so deeply ingrained in my DNA. So I guess that's my answer. It's not a great one
Your
at
answer
that.
is I reject
But
the question,
I
which
reject the you said
I have not had that one before, but
that.
I'm going to answer it for you based on something we talked about earlier. I think if you were born 400 years ago, you'd be sailing the high seas as an
I would
explorer,
be. I absolutely would.
filling out the map.
I would do that.
Because yeah, you've gone into inner space. You've explored inner space now and molecular space. If it were 400 years ago, we'd have a nation called Soragni or something like that.
But good we don't. But yes, I would certainly be exploring in some ways. Yes.
Finally, what's a great piece of advice that you've been given before, either professional or not?
I think this kind of ties back to the things we were discussing earlier. It can feel very hard when you're starting or when you're new or when you're not of the right, social group, or you're not of the right, type of person. It feels very hard to find your place and your voice in science. But despite Twitter not being there anymore and so on and so forth, I think it's so important for trainees to go out there, to listen to the scientists, to put yourself out there, in whichever social platform you choose to do. Those networking opportunities are invaluable, and you can do that in person too, right? Like, never give up an opportunity to go and meet and talk to people. It can be very intimidating. It certainly is, but I think there is so many opportunities for science collaboration and for your personal, advancement that are truly important there. And so, read a lot of papers, talk to a lot of people. I think those things are very important.
That's great advice. Be visible, and in
You got
a
to be
world
visible.
where it seems like so many of us feel unseen. I think it's important to underscore that we are looking for you guys. We're looking for the trainees, and visibility is critical to getting moving forward. Critical for us and critical for you to move the science forward. So thanks for sharing that piece of advice. I should take that advice myself, to be honest.
On social media tomorrow.
Put me out there. Alice, thanks again so much. This has been so fun, and you really bring so much to the table across the board. I don't know. Arun's got his reservation for the space station, going to explore the outer bounds. Maybe there's room for you there too, if you ever get bored in the inner space front.
I wouldn't mind that opportunity. I guess I have another job in mind.
Anyway, terrific chat, and thanks again. We'll have you on soon again.
Thank you so much. Nice talking to you guys.
All right, guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast, or maybe try Blue Sky at STEMCELLpodcast.com, or via email at info@stemcellpodcast.com. At all those outlets, you can give us feedback or suggest guests. Episode 303, closing out. Thank you guys so much for listening.